Hello dear learners, welcome to my channel Embrace Biota. This is the part 3 of our cell biology. In the previous parts we have discussed about cell wall, cell membrane and many of the cell organelles. Today's lecture is about the microbodies and the cytoskeleton. First one is the cytoskeleton. The cytoskeleton is an intracellular network which is present only in the animal cell. It is meant for providing the support, maintaining the shape and help in the organelle transport. The cytoskeleton is a proteinaceous structure so it can assemble or disassemble rapidly. Cytoskeleton is single membranous structure. It has three main components. This framework is composed of microtubules, microfilament and intermediate filament. Firstly, we have microtubules. Microtubules are long, hollow and cylindrical structures. It is made up of protein tubulin. Synthesis of this tubulin protein occurs in the G2 phase of the cell cycle. This tubulin protein is formed by the polymerization of two subunits. These are alpha tubulin and beta tubulin. The alpha and beta forms the dimer. Dimerization gives rise to the network of microtubule. These alpha and beta dimer polymerize and forms a protofilament. In the protofilament, the alpha and beta subunit arranged in alternate fashion, and the one microtubule is formed from the organization of 13 protofilament. The microtubular structure can exist in the three forms. One is singlet, second is doublet and the third is triplet. In singlet, only one single microtubule is present and one single microtubule is composed of the 13 protofilament. So the singlet have only 13 protofilament in it. Second is the doublet microtubule, in which there is one complete microtubule, that is 13 protofilament, is associated with an incomplete microtubule which is composed of the 10 protofilament. So the doublet microtubule consisting of a complete protofilament and incomplete protofilament, that is having 13 protofilament ring plus 10 protofilament ring. Whereas in triplet there is one complete microtubule with which the two incomplete microtubules are associated. That is one complete protofilament plus two incomplete protofilament. It means the 13 protofilament are associated with the two set of 10 incomplete protofilament. After this we have the arrangement of these microtubules. The microtubule shows the two types of arrangement. First one is the 9 plus 2 arrangement and the second is 9 plus 0 arrangement. In 9 plus 2 arrangement, the 9 doublet microtubule is associated with the 2 singlet microtubule. The doublet microtubules are present towards the periphery and the singlet is at the center. So the 9 set of doublet are at the periphery and the 2 set of the singlet are at the center. This 9 plus 2 arrangement of microtubule is seen in the cilia and flagella. Second is the 9 plus 0 arrangement. In 9 plus 0 arrangement, there is 9 set of the triplet microtubule and 0 indicates that nothing is present in the center. These 9 set of the singlet microtubule are towards the periphery. 9 plus 0 arrangement is found in the centriole and the basal bodies. The structure of the cilia and flagella are example of 9 plus 2 arrangement of microtubules. The difference between the cilia and flagella are the cilia is smaller whereas the flagella is longer structure. But the similarity is that both the cilia and flagella arise from the basal body. In basal body there is 9 plus 0 arrangement of microtubules. The cilia is the feature of only eukaryotic cell. So the cilia is the hair-like extension of the plasma membrane that helps in the movement of the structures. It is seen in the paramecium, Botticella and Opelina. In human body, the cilia is present in the trachea and the fallopian tube. Second is the flagella, which also have the 9+2 arrangement just like the cilia. Flagella is the extension of the plasma membrane in both prokaryotes as well as eukaryotes. It is seen in the Euglena, Trypanosoma, Leishmania, dinoflagellates. In human, the flagella is present in the tail of the sperm. The ultrastructure of the flagella shows the 9+2 arrangement. But the flagella of the bacteria do not show this 9+2 arrangement. In the doublet microtubule, there is the interdoublet ridge is present known as nexin. The doublet are 9 in the number, so as the nexin is also 9 in the number. And the singlet is surrounded by the central sheath, which is proteinaceous in the nature. With the central sheath, the radial spoke is attached. These radial spokes connects the singlet to the doublet. And the singlet consists of the dynein arms, which regulates the beating of the singlet microtubule. After cilia and flagella, we have the centrosome. The term centrosome was given by the Theodore Boveri. It is only present in the animal cell and absent in the plant cell. It is present just outside the nuclei of the animal cell. Centrosome comprises of the cytoplasm and centriole. The centrosome contains the organelles known as centriole. These centrioles are always present in the pairs, so known as the diplosomes. These are known as the proximal centriole and the distal centriole. These centrioles are oriented at right angle to each other. The two centriole are found inside a specialized cytoplasm known as the centrosphere or kinosoplasm. The centriole is composed of the 27 microtubules which is arranged in 9 triplets and give rise to the 9+0 arrangement. The central sheath here known as the hub. The structure of the centriole appears like the cartwheel. Each triplet is joined with the nexin. If we talk about the function of the centriole is the structure in the cell which is concerned with the spindle formation during cell division. The centriole have the power of the duplication and the duplication occurs in the S phase of cell cycle. The second component of the cytoskeleton is the microfilament. The microfilament are the long, thin, helically intertwined filament and made up of the polymer of protein actin. Globular protein G-actin polymerizes and forms the filamentous structure known as F-actin. The 2F-actin also polymerize and forms the actin filament. Actin is known for its role in the muscle contraction. So the microfilament plays the structural role and interact with the microtubule and intermediate filament to form the cytoskeleton. Up next is the intermediate filament. Intermediate filament are tough, elongated molecule present in the bundle and is made up of 7 amino acids. The 7 amino acids here are known as the heptad. The two heptad combines and forms the dimer. The dimer forms the tetramer and the further coiling result in the formation of rope-like structure that is intermediate filament. The examples of intermediate filament are lamine, A, B, C and keratin, desmin, peripherin. Lamine A, B and C are found below the inner nuclear membrane and forms the nuclear lamina. After cytoskeleton, the second topic of today's discussion is the microbodies. The microbodies are small organelles which is of following types. First one is the peroxisome, second is the spheroosome, third is the glyoxysome and fourth is the lomasomes. The peroxisome is present in both plant and animals. Spheroosome is seen only in plants and the glyoxysomes are also present in plants only, whereas the lomosome is seen in the plants and fungi. First is the lomosome. Lomosome is the single membranous microbody present between the cell wall and plasma membrane. Its role is the synthesis of the cell wall. Second is the peroxisome discovered by the D. Dewey and Baudin. Peroxisome is present in the plants but in animals it is also seen in the kidney and known as the uricosome. The two important enzymes associated with the peroxisome are urate oxidase and catalase. The peroxisome contains oxidizing enzymes and promote gluconeogenesis in animals. Whereas in plants, in the mesophyll cells of the leaves, the peroxisome interact with the mitochondria and chloroplast. With these two, it takes part in the photorespiration. During the photorespiration, the hydrogen peroxide is formed and is broken down into the water and oxygen with the help of the catalase enzyme. The urate oxidase plays the role in the catabolism of purines. After peroxisomes, we have the glyoxysome. The discovery of the glyoxysome is done by the weavers in the endosperm of germinating cell. It is only present in plant cell. The glyoxysome usually occurs in the fat-rich plant cells associated with the triglyceride metabolism through the glyoxylate pathway. The glyoxysome serves as the enzymatic site for the reactions including the conversion of the stored fatty acid into the carbohydrates. Another important function is the gluconeogenesis and converts the triglycerides into carbohydrates. After glyoxysome, the last microbody is the spheroosome. The spherosomes are similar to the lysosomes, so it is also known as the plant lysosome. The spherosome are derived from the endoplasmic reticulum and contains about the 40% of the fats. Spherosome have high affinity for the fat stain. These are associated with the synthesis and storage of lipids and also possess the lysosomal activity. So this is all about the cytoskeleton and microbodies. If you find this video helpful, subscribe the channel. My name is Anna Sharma and thanks for watching.